Hannah Inforzato presents Physical enrichment enhances memory function by regulating stress hormone and brain acetylcholine esterase activity in rats exposed to restraint stress. This experiment proposes the hypothesis of experiencing an enriched environment from early life may exhibit alterations of endocrine responses to stress, leading to decreased adverse effects of stress in adulthood. Early life experiences can change a person's mental ability to cope with stressful situations. A healthy and stimulated environment can mean positive effects on brain function. Stressful events when one is young can lead to hyperactivity in the hypothalamic adrenal pituitary axis. Increased glucocorticoid levels can lead to impaired learning and memory function. This is done when the glucocorticoids bind to receptors that control the activity of the HPA axis. Physical stimulation and social interaction can decrease the severity of stress. Environmental enrichment affects neuroplasticity and neurogenesis through the production of neurotrophic growth factors. This enriched environment may help decrease accelerated aging due to stress and reduce the risk of memory loss associated with aging. Exposure to repeated stress interrupts the formation of memory and then negatively affects the performance of learning a task. And this can lead to oxidative stress, which is increased production of free radicals and abnormal antioxidant enzyme activity. 24 rats aged 4 to 5 months were collected and given standard food. The rats were divided into 4 groups: control, stress, enriched environment, and enriched environment plus stress. The control and stress groups were placed in standard cages, while the enriched environment and the enriched environment stress groups were kept in bigger cages. The enriched environment groups of rats had small sheltered houses, colorful toys, PVC pipe tunnels, and a running wheel, while the control groups had nothing. The 2 stress groups were exposed to restraint stress and closed plastic tubes 2 hours a day for 7 consecutive days. This treatment was given after 4 weeks of pre-exposure to social or physical enriched environments. The Morris water maze was used to assess short-term and long-term memory before and after stress treatment. The experiment consisted of 3 phases: training, where the rats were allowed to swim around and locate hidden platforms within 120 seconds; short-term memory, where the rats were allowed to find the hidden platform 60 minutes after training; and the long-term memory, where the rats were allowed to find the hidden platform 24 hours after training. Escape latency is described as the time to reach the platform, and a reduced escape latency during tests means an improved memory retention. The rats' brains were collected after the test and dissected to assess the hormone levels. The figure on the left shows the amount of time for the rats to find the hidden platform 60 minutes after training. The rats kept in an enriched environment had considerably lower escape latencies before restraint stress than the rats kept in standard cages. All groups except the control group had an increased escape latency or a decreased memory retention after the restraint stress. The figure to the right depicts the levels of acetylcholine esterase in the brain tissue. Acetylcholine esterase is an enzyme that breaks down the neurotransmitter acetylcholine, which is released when one is under stress. Higher acetylcholine esterase levels are observed in the stressed rats, but the enriched environment rats have overall lower levels of acetylcholine esterase. This data supports the hypothesis and the predictions made in the experiment. The results of this study are consistent with previous studies as well. A change in environment can affect performance as well as biochemical parameters that control these mechanisms, and the implications of this experiment is that children raised in an enriched environment can manage stress effectively in the future.
Exercise is also important in brain function in that it prevents the spread of tumors. The article that I am presenting is entitled "Exercise Maintains Blood-Brain Barrier Integrity During Early Stages of Brain Metastasis Formation." This study was conducted in 2015 by Wolf, Davidson, Roble, and Taborek. These scientists wanted to research how cancer spreads into the brain and whether or not the blood-brain barrier could be strengthened through exercise. In order for tumors to progress into the brain, they must find a passage through the blood-brain barrier, which is a highly protected, multivascular barrier filled with tight junction proteins. The tight junction proteins can be regulated and strengthened under physiological and pathophysiological conditions. The research objective in this experiment was to prove that exercise can minimize oxidation status in the blood-brain barrier and protect against the spread of brain tumors. The question this research asks is whether or not exercise can help prevent the spread of tumors in the brain. The research team hypothesized that with exercise, the brain will not have as much oxidative stress, meaning that the brain can better fight off and prevent the spread of tumor cells. When they refer to oxidative stress, they are referring to the imbalance between the production of free radicals and the body's ability to counteract their harmful effects through antioxidants. More oxidative stress means that there's more damage to the biomolecules in the brain and a greater risk of disease. The researchers predicted that if tumor cells are injected into the brains of mice and one group of mice is exercised for 5 weeks, then the exercised mice will exhibit less brain metastasis than the sedentary mice. In this experiment, mice were split into 2 groups, exercise and sedentary. Scientists infused tumor cells into the brains of both groups via the internal carotid artery and measured the amount of cells once each week via a bioluminescence image and once at the end of the study, as the mice were euthanized and their brains were dissected. The exercise group ran on a wheel every day for 5 weeks, while sedentary group was confined to a cage for the duration of the experiment. The cages were the same in both groups, but the exercise group had voluntary access to a running wheel, while the wheel in the cages of the sedentary group was locked so that they could not run. The independent variable in this experiment was the exercise, and the dependent variable was the size of the brain tumor. The control variables were that the mice were handled the same way in both groups, and they were both subject to the same temperature, noises, food, and light. The mice were subject to a regular 12 hours of light and 12 hours of dark. As you can see by the graphs, the results of this experiment were that the brain tumors spread less in the exercised mice than in the sedentary mice. The graph on the left depicts the mean number of tumor cells as well as the metastatic area in both groups of mice after 48 hours and after 3 weeks. The graph on the right shows the brain tumor bioluminescence in both groups of mice for the duration of the experiment and a comparison at 3 weeks. The brain tumors were both smaller and showed up less on the imaging technology in the exercised mice than in the sedentary mice, thus showing that the brain exhibited less oxidative stress with exercise and prevented the tumor from spreading. Therefore, the hypothesis and prediction of the researchers was supported. The scientists were able to conclude from this experiment that exercise reduces the spread of tumors in the brains of mice. This study is significant because it opens doors for other studies, possibly for humans, that could test to see if human brains would react the same way and possibly increase the lifespan of someone who has cancer in the brain.
Exercise also has an impact on muscle stiffness, creating a difference between athletes and non-athletes. This experiment looked at the influence of athletic status on the passive properties of the muscle tendon unit in traditional performance measures in Division I female soccer players and non-athlete controls. A person's athletic status is used by athletic programs to determine athletic ability and identify prospective members. Many coaches attempt to find individuals with the ability to succeed as a high-level athlete. This experiment examined the muscle stiffness in NCAA Division I female soccer athletes to determine their athletic status compared to non-athlete controls. Soccer athletes typically have strong legs, Because of this, the researchers examined the muscle stiffness in the posterior hip and thigh muscles. The athletes are expected to have stiffer muscles than the non-athlete controls. The researchers also performed a secondary experiment in which they examined vertical jumps from each of the groups and compared the results to find muscle power. They then examined the relationship between muscle power and muscle stiffness. It was hypothesized that stiffness would be related to lower body power output. The researchers examined two groups, Division I female soccer players and non-athletes. The group of non-athletes served as the control group. There were 10 Division I female soccer players and 11 non-athletes. Both groups performed an instrumented straight leg assessment and a vertical jump to test the stiffness, range of motion, and lower body power characteristics of each of the groups. Each participant visited the lab twice with 2 to 3 days separating each visit. In the first visit, the participants performed vertical jumps and instrumented straight leg assessments. In the second visit, the participants performed counter-movement vertical jumps and passive instrumented straight leg assessments. The passive instrumented straight leg assessments, also known as ISLR, consisted of patients laying in the supine position with the knee in full extension and the ankle at a 90-degree angle. The leg was then placed into a custom-made cast to keep it in this position. All analyses were done on the right leg in order to keep consistency. There were no statistical differences between the two groups regarding age, height, mass, and muscle, which made the comparison from the results fair. After the tests were performed, there was no significant difference in the range of motion between the female athletes and non-athletes. However, the athletes were found to have higher lower body power based on the results of the vertical jump. Since the researchers were able to distinguish a difference in lower body power and muscle stiffness between the athletes and non-athletes with the same body measurements, they are able to make the determination that there is a difference in athletic status in college-aged females. There was also a significant difference in the lower body power output, as shown in the results from the vertical jump. This difference correlates to the difference in muscle stiffness, which supports the idea that muscle stiffness plays a key role in determining athletic status. Since the athletes and non-athletes' muscle ECSA was similar, the difference in muscle stiffness could be a result of training-induced qualitative factors. Additionally, the difference in athletic status could be a result of the large amount of training that the athletes perform on a daily basis and the body's ability to adapt to this work. Overall, the results of this experiment supported the hypothesis that athletes will have a higher muscle stiffness than non-athletes and that stiffness is related to lower body output. Given the fact that muscle stiffness correlates with athletic status, coaches can use this information as a means of performance evaluations for recruiting athletes. Also, the study of muscle stiffness can be used to find a way to reduce knee injuries such as those concerning the ACL.